Good evening, everyone. I'm Xiaoding, working at Sun Yat-sen Memory Hospital. Today, I would like to present one of our recent work, nanoparticles mediate non-coding RNA silence for effective cancer radiotherapy. So because Pierre has introduced me, so I will not introduce myself. So next I will go into my talk. So as we know, more than 19% of our human genes cannot encoding proteins, but can be transcribed into non-coding RNAs. These non-coding RNAs may export their biological functions. Currently, it has been demonstrated that many non-coding RNAs can evolve intracellular signal networks and regulate the development and the progression of tumors. For example, the long non-coding RNA HTAL can promote the invasion and metastasis of breast cancer and lung cancer through the activation several classic signal pathways such as Wnt beta-catenin pathway and NF-kappa B signal pathway. Therefore, explore new functional non-coding RNAs and understanding their regulation mechanisms could provide new therapeutic targets for effective cancer therapy. So long non-coding RNA are class of long non-coding RNAs are class of non-coding RNA with a length more than 200 nucleotides. Link RNAs are widely involved in tumor genesis and progressions, and the regulation of their expression usually induce multiple biological functions. In general, long non-coding RNAs can interact with chromatin, or interact with protein or RNA to regulate protein expression or regulate gene expression. Currently, many long-coding RNAs has been discovered, and they have been widely involved in tumor genesis and progression, such as promoting tumor cell proliferation, evading apoptosis, and improving angiogenesis. Due to their important functions, link RNAs has been now identified as attractive therapeutic targets for effective cancer treatment. And also now several clinical trials based on regulating link RNA functions are now ongoing. So radiotherapy is an effective treatment for many cancers, but radio resistance is frequently encountered in clinic. Ionized radiation can induce the generation of ROS, high-level ROS, and this ROS could cause DNA breaks or activate apoptic signal pathways to induce cell apoptosis. However, tumor cells usually adaptively generate a high concentration antioxidants and reduction agents such as GSH, which can scavenge the ROS and maintain the intracellular redox homeostasis for cell survival, and finally inducing radio resistance. Therefore, disrupting intracellular redox homeostasis through promoting ROS generation and/or reducing ROS scavenge has been long studied as an effective strategy to enhance radio sensitivity. However, radio resistance is a complex process associated with multiple biological changes that are regulated by intracellular signal pathways. Therefore, benefit treatment for radio-resistant cancer patients needed to include comprehensive consideration of multiple biological changes contributing radio resistance. So based on our formal consideration, we speculate that there are some link RNAs to involve in radio resistance, and explore the key link RNAs would facilitate to reverse radio resistance. In addition, due to the generation of a high concentration of reduction agent, such as GSH under radiation, systemic delivery of siRNA using reduction-responsive nanoparticles can synergistically reverse radio resistance through concurrently silence link RNA expression and scavenge intracellular GSH, leading to effective cancer radiotherapy. So along this principle, we in this work we collect the matched tumor tissues of 2 triple-negative breast cancer patients. Also, we call it TNBC patients before post-operative radiotherapy and after post-operative radiotherapy with local recurrence, and then employed link RNA microarray to analyze and compare their link RNA expression profile. In addition, we also construct radio-resistive triple-negative breast cancer cells and compare the link RNA expression file with the parental cell line. Finally, we found that 6 link RNAs are upregulated in both radio-resistant tumor samples and cells. We also use qPCR analysis, and the result shows that the link RNA AFAP1 and AS1 is highest in the radio-resistant triple-negative breast cancer cells. So to further validate the high expression of link AFAP1, AS1 is to further examine the high expression of this link RNA in radio-resistance TNBC. So we next this link RNA expression in 12 TNBC patients received post-operative radiotherapy. It can be seen in the figure compared the patient without recurrence. Higher link AFAP1 AS1 expression can be found in the tumor tissues of the patient with recurrence. In addition, through examining the link AFAP1 AS1 expression in 125 TNBC patients, we found that its high expression is associated with poor DFS and poor OS. In addition, in this 125 patients, there are 22 patients with local recurrence, and in these patients with local recurrence, high link AFAP1 AS1 expression is found. So we can from this data and conclude that the high AFAP1 AS1 expression is associated with the radio resistance of TNBC patients, and the expression level of this link RNA in tumor tissues could be used to predict the outcome of radiotherapy. So we next examine the intracellular location of this link RNA. We use the FISH, and the result shows that the most of the link RNA is located in the cytoplasm. Then we silence the link AFAP1 AS1 expression, use the lipofectomy, and the result shows that this link RNA silence can reverse radiosensitivity of radio-resistant cells, including low cell viability and more apoptosis. However, if overexpression of the link AFAP1 expression, the cancer cells show the radio resistance. So next, we want to explain why link AFAP1 overexpression induces radio resistance. So we used iron pull down and then use mass spectra to identify the protein interacting with link AFAP1 AS1. As shown in this figure, beta-catenin is the specific link RNA binding protein. This specific interaction is further demonstrated by the RNA pull down followed by Western blot analysis. In general, beta-catenin expression is tightly controlled by the destruction complex, in which the tumor suppressor protein axin act as a scaffold to interact with beta-catenin, APC, CK1, and GSK3 beta. In this destruction complex, CK1 and phosphorylate GSK3 beta can induce the phosphorylation of axine-bonded beta-catenin, leading to beta-catenin degradation. Therefore, we predict that link FAP1S1 might suppress the function of the beta-catenin destruction complex, and thus induce the release of beta-catenin to play its biological functions. To verify our prediction, we silence the link FAP1S1 expression and examine the expression of CK1, GSK3 beta, and the phosphorylation of GSK3 beta and the beta-catenin. We found that high link FAP1S S1 expression show low level of GSK3 beta phosphorylation and high expression of total and active beta-catenin. After silence this link RNA expression, there is no obvious in the level, no obvious change in the expression of CK1 and GSK3 beta. However, the phosphorylation of GSK3 beta is obviously improved. So this result indicates that the link FAP1-AS1 can block the function of beta-catenin destruction complex by suppressing the phosphorylation of GSK3 beta expression, thereby leading to the release of beta-catenin and activating the classic Wnt beta-catenin signaling pathway to promote breast cancer cell proliferation and migration. So we further examine the dissociating constant between link AFAP1-AS1 and beta-catenin. We added different amount of this link RNA and then used the best plot to analyze the expression of beta-catenin. And from this figure, we can find that the KD between this link RNA and the beta-catenin is around 3.59 nanomole. So next, because link AFAP1 can activate the classic Wnt beta-catenin signaling pathway to induce the radio resistance. So we guess that, so we think that systemic delivery siRNA to silence this link RNA expression could be a promising strategy to reverse radio resistance. So we previously developed a series of nanoparticles for systemic siRNA delivery, just like Professor Jin Junshi reported, including redox responsive, pH responsive, RS responsive nanoparticles. But considering the generation of a high concentration of a reduction agent, GSH, and radiation, so we chose reduction responsive nanoparticle for siRNA delivery and expect to synergistically reverse the radio resistance through the concurrently silence the link AFAP1-AS1 expression and scavenge the intracellular GSH. So this reduction responsive nanoparticle platform is composed of reduction responsive polymer, we call it PDSA, and outer PG shell, which could prolong the blood circulation of the nanoparticles. As shown in this figure, after loading the link AFAP1-AS1 siRNA, the nanoparticle shows uniform morphology with a size around 110 nanometer. Use this nanoparticle to treat the radio-resistant breast cancer cells. More than 70% of the link RNA expression could be suppressed at siRNA dose are around 30 nanomole. With this efficient link RNA silence, beta-catenin expression is obviously suppressed. More importantly, because these nanoparticles are reduction responsive, they can scavenge the intracellular GSH, and thus the GSH expression always maintains a low level under radiation. With this link RNA silencing and the GSH scavenge, the nanoparticles could synergistically reward the radio resistance of breast cancer cells. As shown in this figure, the nanoparticles are the most effective in inhibiting the survival of the breast cancer cells on the radiation compared to the single GSH scavenge or single link RNA silencing. So with the above promising in vitro experimental data, so we finally evaluate whether the reduction responsive nanoparticle can deliver link RNA, siRNA, and enhance the therapeutic outcome of radiotherapy. So to this end, the nanoparticle was intravenously injected to the breast cancer cell xenograft tumor-bearing mice once every 2 days. After 3 consecutive injections, the tumors show favorable sensitivity to the radiation, and the tumor growth is obviously inhibited. As we know, radio resistance usually leading to enhanced local invasion and metastasis of cancer patients. Therefore, in this work, we established a metastatic model using the luciferase expression radio resistant breast cancer cells to further demonstrate the ability of our reduction responsive nanoparticles for siRNA delivery to enhance the radiotherapy effect as we expected, with the concurrent link AFAP1S1 silencing and GSH scavenge, the nanoparticle treatment followed by radiation shows the strongest ability to reduce the metastatic tumor burden. So a brief summary. So in this work, we have identified a functional link RNA. We call it link AFAP1S1 in radio-resistant TNBC patients, and this link RNA could induce radio resistance where activating beta-catenin signaling pathway. Silencing this link RNA expression using reduction responsive nanoparticles could synergistically reverse radio resistance. I would like to thank the support from several grants such as the NSFC and the 1000 Talent and the Department of Science and Technology of Guangdong Province and the Guangzhou Science and Technology Bureau. And also this work, we collaborate with Professor He Yao and Professor Hu Hai and also Professor Pierre. And also I would like to thank the students, Dr. Zhou Feibi and Dr. Qingjian Li. Finally, thank you for your time to listen to my talk. Thank you again.
Yeah, thank you so much, Xiaoding. I think it's hard for Xiaoding actually to talk in English right now because we have been back for maybe 3 years now. I think it's, yeah. So Xiaoding has not been presenting in English. So we really thank you for today. And so this is a classic example of really an integration, a really good integration, because before we came back, I think Xiaoding was working on polymeric nanoparticles, but not in significant way of integration with the clinics. But when we come here, when we arrive at Sun Memorial Hospital, and we found out that actually we are the pioneer of long known coding RNA, which Professor Xiaoding has mentioned. So knowing that and the basic research that they have done, and then Professor Xu just took it to another level. So he took the link RNA and then encapsulate in the nanoparticle, and then now we try to deliver this to solve a clinical problem that is radio resistance. So I think this is a very classic example of why we should